Thank you. Good evening, everyone. My name is Shoni Mulibana. I'm a pharmacist. I've been working for an infectious diseases organization that supports government and other structures in the rollout and implementation of the HIV, TB, and other related diseases to make sure that we treat health seriously. I'm also one of the co-opted members of the TEASA health desk, if I may say so. Nombuso will share my presentation. While she is sharing my presentation, I'll just echo Prof. Muzi's sentiments that basically he has done half my presentation. I don't even, I think a lot of things have been explained from what he has indicated. I'll just quickly go through vaccines just to explain what a vaccine is and how it differs from medication. And then we would look at different types of vaccines that are available and how vaccines are developed and registered and approved for registration. And then we will look at the COVID vaccines that are already available. And at the end, we'll just touch briefly on why there is, people are asking why the COVID vaccines have not taken longer than the other vaccines. Okay, and next slide.
Okay.
I don't know if I will just raise my voice because there are people who are indicating that they cannot hear. So a vaccine is a biological preparation that improves immunity to a particular disease. So this is it's more like a like cures like situation with vaccines, where the vaccines, you take the very specific antigen that causes disease and use it to cure or prevent that disease. So those organisms, they are usually made from the weakened or killed forms of the microbes or the toxins or one of its surface proteins. It stimulates the immune system to produce antibodies, exactly like it would if you were exposed to the disease. So it mimics the disease itself. After getting vaccinated, you develop immunity to that disease without having to get the disease first. And then the immune system quickly clears the germs from the body so that they don't cause the diseases. And your body in turn remembers that germ so that later in life, when you get the disease, then they are able to recognize and prevent that disease. So there are various types of vaccines that are available. We have a live attenuated vaccine. So these vaccines contain a modified live germ that has been weakened. So it's more like the organism that causes the disease, but it has been weakened so that it doesn't cause the disease. So it stimulates the immune system in a similar manner to the pathogen and elicits a strong immune response. And often confer lifelong immunity with only 1 or 2 doses. A good example would be the yellow fever vaccine. If you only need 1 or 2 doses for that, and then you don't have to be vaccinated again. They can be easy or difficult to develop, specifically the ones that are created for bacteria, like BCG. So we will talk about later on. You will see the process that goes into developing vaccines. With bacteria, it's difficult because bacteria have thousands of genes and are much harder to control than viruses. Scientists working on a live vaccines for a bacterium, however, might be able to use recombinant DNA technology to remove several key genes. I think this alludes to what Prof was saying regarding the DNA and the messenger RNA technology. So this approach has been used to create a vaccine, for example, the one that we use for cholera. So the issue of vaccines development, it's not really a new thing. I think now it's just that there is a lot of attention that has been focused on COVID, and there's quite a lot of information that is available. Hence, people think that it's a new thing. This has never happened before. So then, even the COVID vaccines are following what has been done previously, and almost all of us here have been vaccinated before, and our we have not changed. We have not been coded. So to speak. The next slide. So the second type is the inactivated vaccine, which is made by killing the disease-causing germ with chemicals, heat, or radiation. So this is done at a laboratory. Such vaccines are more stable, but they may stimulate a weaker immune response than do live vaccines. So it would take several doses or booster shots to maintain the person's immunity. Inactivated vaccines, usually they don't require refrigerations, which become much more easier for people who are in poor settings, especially because you are going to need many more doses or a booster dose after getting that first dose. There's also a subunit vaccine. This type of vaccines include only the antigens that best stimulate the immune system. Once scientists identify the most appropriate antigens to include in a subunit vaccine, they can either grow the microbe in the lab and then use the chemicals to break it apart and gather the important antigens or manufacture the antigen molecules from the microbes using the recombinant DNA technology. Vaccines produced this way are called recombinant subunit vaccines. Next slide. We also have the Toxoid vaccines. These vaccines are made by inactivating toxins by treating them with formalin, a solution of formaldehyde and sterilized water. Such detoxified toxins are safe for use in vaccines, and they are made for bacteria that secrete illness-causing toxins or harmful chemicals. When the immune system receives a vaccine containing a harmless toxoid, it learns how to fight off the natural bacterial toxin which causes an illness. And some of the vaccines that use this type of technology are vaccines against diphtheria and tetanus. That's why you have a tetanus toxoid vaccine. Next slide. The conjugate vaccines, these vaccines, they link polysaccharides to a carrier protein antigens or toxoids from the same microbe. They disguise a bacterium's antigens so that the immature immune response systems of infants and younger children cannot recognize or respond to them. The example for this type of a vaccine is the ones that we use for Haemophilus influenzae type B. DNA vaccines, once the genes from a microbe have been analyzed, scientists could attempt to create a DNA vaccine against it. Still in the, most of these are still in the experimental stages. However, they are showing great promise. I think the development of COVID vaccines has shown these experiments that have been going on for a very long time. So as much as it looks like we are having a vaccine within 10 months, some of these experiments have been happening for a very long time. And now they have just accelerated the development. And because there has been quite a lot of investment made into the development of COVID vaccines, there has been fast tracking of some of the phases and some of the research and development that made it possible for these vaccines to look like they have been done in a very short space of time. And also the use of technology. I think technology has improved quite to do in months. We can get or do in weeks. I think with technology, we know now the world also has become quite very small. Previously it would take us a long time to get to a certain place or to get news from a certain place. But now things as and when they happen, we are able to get that. So the technology advancement has also assisted in terms of moving the research and development forward. Next slide. So the last type that I'm going to talk about is the recombinant vector vaccines. So the vector refers to the virus or bacterium used that is used as a carrier. These are also experimental vaccines similar to DNA vaccines, but they use an attenuated virus or bacterium to introduce microbial DNA to cells of the body. They also mimic a natural infection and therefore do a good job of stimulating the immune system. Scientists are working on both bacterial and viral-based recombinant vector vaccines for TB, HIV, rabies, and measles. So you can see that already with the advancement of COVID, we are moving forward. So this picture shows the various stages of vaccine development and the regulatory process. So the first stage is exploratory phase, where the scientists are really exploring the feasibility of a vaccine. And then this is where regulation, they regulate as they go. This stage involves basic lab processes, research, and usually it would last for 2 to 4 years. And then mostly facilities that explore the feasibility of having either it's drugs or vaccine are federally funded, either pharmaceutical companies or academic institutions. They would be funded to look at the possibility of having a particular vaccine. So this is where they do a proof of concept, and then it gets validated. And once they are quite sure that this will go, it goes into the preclinical stages. So the preclinical stages, that's where they use animals to test the safety of drugs, and also to see the immunogenicity of a vaccine, if this vaccine would really create an immune response. Once the preclinical stage has been done, there is an application process that happens. So the company or a sponsor would apply for what we call innovative new drug application. So they would write a protocol that explains the processes, explains the laboratory processes that they're going to do. It explains safety measures. It explains the efficacy of what is expected to come out of the drug. It also explains what will happen should there be any side effects. And this goes to, in South Africa, it will go to SAPRA if there is a company that is innovative or that is developing the drug. But most of these innovation companies are in the US, so they would apply to the FDA, the Food and Drug Administration, that would then approve that the clinical phase can happen, the clinical trials can happen. So the clinical trials that's where now with phase 1, we start with human subjects. Many vaccines don't really pass through the preclinical stages because they are found that they don't really generate that immune response that is required. So most of them are abandoned, and it is a standard process. It is known, it is also expected in some cases. So we have now already heard of one a vaccine candidate that has been abandoned. So it doesn't necessarily mean that vaccines don't work, because there is that one candidate vaccine that has been stopped. That is part of the process. So phase 1 involves a small group of people, about 20 to 80 individuals, to determine the safety of the drug or safety of the vaccine. And then if phase 1 proves that the vaccine is safe, then there would be a regulatory process. So each and every step, there is a regulatory process. You are approved to move to the next phase. The company doesn't just decide that, no, we have tested this vaccine on 80 people, it is safe, and then move forward. There are regulatory requirements that they need to that they are subjected to, and they need to stick to according to the protocol. Phase 2 involves about several hundreds patients. So this one, it was to determine the immunogenicity of the vaccine as well as the safety of the vaccine. So it will determine the optimal dose, scheduling of the vaccine. If you're going to have 1 or 2 doses, will it require a booster, issues like that. And and because then it's a little bit more patients, then you do your safety assessments. And in this phase, then they can start taking also looking into the target groups, people who have the condition, to see if it will work for those people. So if phase 2 passes and it is approved that the drug is safety, it generates an immune response, or on this smaller, a little bit larger population, then we move on to phase 3 of clinical trials, where here it involves a larger group of people. This is about tens of thousands. For instance, the Johnson & Johnson trial that was also conducted here in South Africa, they needed to do phase 3 trials on about 60,000 people in different areas. So this, with phase 3, they are testing the safety, the efficacy, and also the quality of the drug, of the vaccine. And then you look at the target population. Once phase 3 is complete and the drug has been proven safe, the vaccine can be registered with a regulatory authority. So the registration process is the company needs to prove that the drug is of good quality, it is safety, and it is efficacious. And then they will do evaluations of risk, the risk-benefit ratio, and there has to be a balance regarding that. So the development process takes into consideration the individuals, the human rights, because previously, if you look at the history of drug development and medicine usage, in the beginning, where they didn't have regulations, there were issues that happened, and human rights were not taken into consideration. So these processes are quite stringent currently, because we need to look into that. So we are quite satisfied that the process that is being followed for all the vaccine candidates currently are going to protect our population and the communities. Thank you.
Thank you, Mulibana. We are clapping hands virtually as well. Thank you for that important enlightenment. enlightening, sorry. Colleagues and the future speakers, if you see me showing my face, it means the time is now up. Your allocated time is now up. So please bear that in mind.